All right. Well, thanks everybody who's here live, and I know there's lots of folks out there who are planning to watch the recording. I appreciate everyone's interest in helping out with our coaster sampling program. The purpose of the call today is to just provide some background on the effort, what we're doing, how we're doing it, why, that sort of thing, and then provide some training on actually taking the samples, which is quite simple. Um, so before we get started, there's a few folks from our project team here on the call, and I'd like, do a quick round of introductions of the folks on the project team. So my name's Jake Lemon. I'm a field and research manager for Trout Unlimited, based in the Lower Peninsula of Michigan, but coordinating our work around coastal brook trout in the Lake Superior watershed. I've been with TU for about 14 years now, working on a variety of monitoring and research related work during that time. So, Nadia, do you want to jump in?
Yeah, sure. Hi, everyone. I'm Nadia Mabuzadeh. I'm a faculty member at North Carolina State University, which you might be wondering how in the world is NC State involved in a project for brook trout in the Lake Superior Basin. I was previously in East Lansing, Michigan, working at Michigan State University for about 6 years on brook trout genetics projects in the Lake Superior Basin. And so we're continuing some of that work here and now North Carolina, which is exciting and fun. So excited to be here and see you all.
Thanks, Nadia. And Troy?
Hi, I'm Troy Zorn. I'm a research biologist with the Fisheries Division. Um, I've worked on brook trout and stream trout in Michigan for 30-ish years. I've been with the division for 38 years, and I've been working on coaster-related projects for about the last 15. We had a Lake Superior wide, couple Lake Superior wide projects, and so this is kind of building off some of that and partnering with Nadia and Jacob and TU and all of you anglers on it. So I'm excited to work together on this.
Thanks, Troy. Paul, are you out there? Doesn't look like it. All right, that's okay. We can forge ahead. So, I have some slides I'm going to share with everybody today, and there'll definitely be opportunity for folks to ask questions. You can either ask questions in the chat or using just by unmuting your speaker or your audio and asking them verbally once the presentation's over. And Troy and Nadia, feel free to jump in on with questions if you feel the urge, or if there's anything that I'm covering that you want to clarify, don't hesitate to jump in. So I think the presentation should only take 20, maybe 25 minutes or so. And then we can jump into some Q&A and talk next steps. So I will share my screen here. All right, you should be seeing my screen.
Looks good, Jake.
Okay, let me get my Zoom controls back up so I can make sure I'm admitting people as they come in here. Okay, so here we go. Um, Still getting set up here a little bit. There we go. All right. Oh, there's Paul. Hey, Paul, you just joined in. We were having some folks from the project team introduce themselves, and you came in right before I was about to start my slide. So can you introduce yourself real quick, Paul?
Yep, sure can. Paul Piszczek, Wisconsin Department of Natural Resources. I'm stationed in Superior, and my management area is the Lake Superior tributaries from their mouths upstream to the first impassable barrier.
Great, thank you, Paul.
You're welcome.
All right, so jumping in here. So coastal brook trout. Well, first off, what is a coastal brook trout? It's make it clear that it's not a separate species. It's a variation in life history expression. So basically it's any trout, brook trout that chooses for whatever reason or to spend part of its life in Lake Superior. These can be there's documented coastal populations that spend much of the year in the lake and then run up tributaries in order to spawn. There's documented populations that actually spawn on shoals in the lake itself and likely probably live their entire life within the larger lake. There's some areas where it's apparent that these fish are moving out of warmer tributaries during the warm summer months into the colder lake for sort of a thermal refuge use. There's some that migrate into the lake in the winter for foraging. There's lots of different things that can make a coaster. And really, what it boils down to are brook trout using the lake for some part of their lifespan. And this can lead to these brook trout growing to larger sizes, as you see, an example here in the picture. As a rule, coasters aren't defined by their larger sizes, but those larger sizes are achieved by it more often by these fish that are using the lake. Um, these brook trout, they exhibit what's called phenotypic plasticity, which is basically they'll change their behavior based on environmental conditions. They aren't locked into a single set of life history behaviors. And so that's why we see these different ways in which they use the lake. And coasters have been in decline for a long time. All the way back in the mid, 1870s, there were already accounts. These are newspaper clippings and clippings from other resources. Way back in the 1870s, there were already accounts of coasters being fished out in certain parts of Lake Superior. And it's the sort of standard suite of issues that contributed to the decline of coasters. One being habitat degradation, the massive deforestation that occurred across the Great Lakes and subsequent wildfires and the sedimentation to streams and overall habitat degradation. Um, Also overharvest. There's some interesting newspaper accounts that have been dug up. For example, over 2 days last week, there were 929 brook trout taken out of the Pilgrim River. On Monday, Mr. Sheldon and two others caught 720 brook trout in the same stream. This wasn't catch and release, so we had a lot of harvest going on. And these two things in concert really contributed to the rapid decline of coaster brook trout and brook trout in general in the Lake Superior Basin. Some photos here showing the extent of habitat degradation, some of the massive logging that took place in the region. This one on the right, sort of the famous walking trees photo, just showing how much erosion occurred following the logging era. So basically you can expect that the sediment level used to be sort of at the base of the tree, but now it's been eroded to the point where much of the root ball is exposed. So, their coasters were really knocked back by these two things, in addition to other types of development, dams, probably logging roads and railroads had contributed to decline. And then subsequently, the introduction of non-native salmonids certainly likely contributed to the ongoing decline of coasters into the 20th century. So recognizing that coasters were in, had gone through a rapid decline or a much lower level than historic records indicate, the Great Lakes Fishery Commission developed what they call the Brook Trout Rehabilitation Plan. So the Great Lakes Fishery Commission, it's a binational commission made up of representatives of the U.S. and Canada and tribes, and it has basically members that work together to try to jointly manage the Great Lakes. And so the commission has technical committees for each lake. The Lake Superior Technical Committee being the one that is responsible for much of the science around fishery rehabilitation and coasters being part of that. And so much of the work in this effort and much of the work we're doing is sort of under the umbrella of the Lake Superior Technical Committee, but actually being implemented by its members, such as the various DNRs and university partners and agencies and whatnot. So when this plan came out, they came forward with some objectives, including creating self-sustaining populations that are geographically widespread. They had some goals in regards to the number of age classes, and they wanted those populations to exhibit genetic profiles found in the Lake Superior Basin, and ultimately to support a managed fishery. And so this 2003 rehabilitation plan really catalyzed a lot of great research and focus and effort around the rehabilitation of brook trout. A lot of research came out in that sort of 2000, I don't know, 5 or 6 to 2012 time frame on the basic biology of coasters and their behavior and various their genetics and whatnot. And a lot of really great work was done. And part of that plan, the folks who wrote it came up with sort of their best estimate of the historic distribution of coasters around the lake. And so you see these dots here. And these came from a variety of sources, from agency paper records, biologist knowledge, library archives, and whatnot. But in reality, they knew they were likely much more distributed, but these were locations where there was some documentation. Um, in recent years, there's been various efforts to document the current extent of coasters around the lake and their current distribution through various means, from genetic approaches and otherwise that we'll talk about here. And those have led to the documentation of some new like potential coaster habitats around the lake. So the red dots were the ones that were in the 2003 rehabilitation plan, whereas the yellow dots are areas where recent research has shown there may be coaster populations. But there's a lot more streams than have been assessed around the Lake Superior watershed. These red dots represent all the perennial cold and cool water streams. So there's 535 of them. And a subset of them have been assessed to date. So this has led to the need for a broader, more integrated assessment of coastal brook trout along around the lake. So in Minnesota, the DNR kicked off what they call their Coaster Genetics Project a few years ago in partnership with Michigan State, and Nadia was one of the researchers who worked on that project. And they integrated their professional staff surveys with collecting fin clips from anglers to build a genetic profile of brook trout in their tributaries to Lake Superior, and to identify what streams might be sourcing coaster brook trout, might be sourcing brook trout that are moving into the lake and being caught in the lake. And this effort was very successful. I'm going to show some of the results of that here in just a second to give you an example, but it included a combination of DNR conducting stream electrofishing surveys and taking fin clips as they conduct those. Also shoreline electrofishing surveys. So this is doing electrofishing in shallow shoreline areas. Same concept as stream electrofishing, basically temporarily stunning the fish so that you can identify and measure and take tissue samples and whatnot. But they also relied heavily on anglers to collect fin clips from the coastal brook trout that they caught in the streams and lakes along the Minnesota shore. And in fact, they said that most of their larger brook trout that were captured did come from their angler volunteers rather than the electrofishing work that was done. And they did a pretty much statewide along the shore assessment of streams along the Minnesota shoreline. And according to what they found, pretty much where they looked, they found brook trout in a lot of places where they maybe didn't expect them or didn't know that they existed. And the methodologies they were using used genetic tools in order to identify the sources of brook trout that were captured in the study. So in this figure, what you're seeing is each dot represents a location where fish were captured. So the dots up here are above barrier. So in most cases above a waterfall that's an impassable barrier. The dots at the lower part along the shoreline are reaches of stream that are below those barriers. And then these were fish that were captured out in the lake. And so for each capture location, using these genetic tools, we can assess where these fish actually came from. So you see in these upper reaches, these are populations that are fish that were born in that stream, have lived in that stream, and were captured in that stream. Whereas some of the below barrier populations, you get a mix. You get fish that are from the stream in which they were captured, and then fish that are from other streams along the Minnesota shoreline. So by seeing these little, different colors within the pie, what we can infer is that, fish were born somewhere in another stream, used the lake to move into the lower portion of the stream in which they were eventually captured. So this basically shows movement of coasters from stream to stream using the lake. Whereas here in the lake, you see a mixed bag of different source streams showing where the fish that were captured were coming from. And what this allows you to do is to start painting a picture of movement around the lake. So you can see these arrows kind of telling us where fish were born, where they're moving to, what fish are moving from stream to stream, from where they're moving out into the lake. And this provided some really useful insights to the Minnesota DNR and to their partners as to what streams are likely sources of coastal brook trout. And really what they found were that those fish were coasters weren't a single population of fish that existed out in the lake. They were more closely related to in-stream populations and even very closely related to populations that exist above impassable barriers. So in many cases, those coastal brook trout may be fish that, were even, born above the barriers, found their way below the barrier, and then moved out into the big lake, potentially due to habitat limitations in those lower reaches. And they were able to really hone in on some watersheds that are these critical sources and, you can focus on coastal brook trout rehabilitation and conservation efforts that are benefiting those in-stream populations while setting them up in the best way possible to support an in-lake coaster fishery. So some really exciting insights that came out of that study and that we are hoping to replicate and see what we can find here in the southern shore of Lake Superior. So another sort of emerging tool or a newer tool for coastal brook trout assessment is the use of stable isotopes. So at a very, very basic level, when fish feed in different studies have shown that the ratio of certain stable isotopes like carbon-13 are different in tissue of fish that feed in Lake Superior versus fish who primarily feed in streams and rivers. So a fish that lives in Lake Superior will have a slightly different, ratio of these stable isotopes than a fish that is found in a stream and is feeding in a stream. So this can help us understand, when we capture a large brook trout in a tributary to Lake Superior, is that a coaster? Is that a fish that's moving in from the lake for whatever reason? Or is it just a large stream resident brook trout? Well, the stable isotopes is a tool that can help us, get at that question. And so if you look at these ratios, you see some groupings of different colors. So we have Iron River here that's sort of grouped over in this part of the graph. Pilgrim River kind of grouped around here. And then we have a grouping here that consists of, Lake Splake, Lake Trout, and some brook trout. So Lake trout being the black dots, Splake being the gray dots, and brook trout that were captured in Lake Superior being the white dots. So we have more of a lake signature here, more of a stream signature over here. But we do have some stream fish that exhibit a lake stable isotope signature. So we have fish captured in some tributaries to Lake Superior that show that stable isotope signature indicative of a lake. And so while these fish were captured in a stream, we can infer that, there's a good chance they've spent some time in the lake. So these genetic tools and the stable isotope tools allows us to take fin tissue samples that were collected from streams and rivers, as well as from the big lake, from Lake Superior, and really start to map out, where these coasters are coming from, where they are going, what are the sources and say what are the sources for these coaster populations or individual coasters, and really dial in a better understanding of just a basic understanding of where these fish are on the landscape so that we can more effective, and that information is just totally essential to effectively managing and protecting and conserving these unique fish. So based on the success of what has occurred up in Minnesota, also in Ontario, they've conducted similar research and used it to inform how they manage their coaster fisheries. We're looking to bring the Coaster Genetics Project to southern Lake Superior along the Michigan and Wisconsin shoreline. And so we've already kind of started to kick this off with we've already kicked this off with both TU and agencies conducting electrofishing assessments on some tributaries of Lake Superior in Wisconsin and Michigan. That's been taking tissue samples, which has been done over the past few years, with plans to scale that up this field season and the following field season, with the goal of hitting hopefully all or as many of those tributaries as we can with via electrofishing. There's also been some amount of shallow, lake electrofishing of some shallow lake areas to capture brook trout and collect tissue samples from in-lake fish. And both of these methods have helped us begin to build a library of tissue samples across southern Lake Superior. But there are certainly gaps that we see in the data set, and gaps that we think anglers might be best suited to fill, especially seeing the success of the program up in Minnesota. So these include just the number of lake caught brook trout that we have in the data set. These fish, especially where they exist in low densities, are really difficult to sample effectively with shoreline electrofishing. Takes a lot of effort to get out there and sample these areas. And so we know that anglers are out, fishing these shoreline areas, probably targeting other fish species, but running into coasters here and there. And so we're hoping to connect with those anglers to help us collect fin clips that will contribute to this research and ultimately contribute to management of these fisheries. Similarly, larger brook trout from our targeted tributary sampling areas are often missing from the data set. And this can be for a variety of reasons. In some cases, these fish may be only moving into those tributaries during certain parts of the year, and that may not be when we're out conducting our stream electrofishing. It could be that especially in the lower reaches of these rivers, it's just not conducive to stream electrofishing due to depth or other factors, and so that we have to conduct our assessments further upstream. But basically, what it comes down to is that anglers are capable of effectively sampling low density fish in some of these areas that we can't get to well and can have a major added benefit to helping us develop a robust data set for this study. Let's see, moving on. So what we're looking for is just a small fin clip. We have a picture here as an example, about dime size from the ventral fin of any brook trout that is caught in Lake Superior off the Michigan or Wisconsin shore. If there's anybody out here on the call that's a Minnesota, fishes more in Minnesota or Ontario, they're doing similar work there. I would need to connect you with a different coordinator for the Minnesota or Ontario project. So let me know and I can do that. But for the purposes of this project that TU and our partners are administering, we're looking along the Michigan and Wisconsin shoreline. So any brook trout, doesn't matter the size, we're looking for fin clips. Also, we're looking for fin clips from any brook trout that is 7 inches or larger that is caught in a lake accessible stream reach. So this is any stream reach that's below the lowermost waterfall or dam. If you need help assessing whether the places you fish or the places you intend to fish in these lake accessible stream reaches, I can help you figure that out. But again, we're not looking for larger brook trout caught in a beaver pond in the headwaters of a river, but we're really looking, lower down in the watershed, again, below these lowermost barriers where fish may have access to the lake. So these fin clips will be collected. They'll be sent to Nadia's lab at NC State, where they'll do the genetic analysis. A portion of the sample will also be sent to a lab at the University of Wisconsin-Milwaukee, where the stable isotope analysis will be conducted. And this will help us build towards some of the output, the map, the data outputs that I showed earlier, helping us understand, again, the sources and locations of coastal brook trout across the southern lake shore. So TU will be providing all the the necessary training, which is this is part of that, the necessary supplies, which I'll talk about in a second, and you'll be shipping shipping those out on a regular basis. And we see this program going for 2025, 2026, and potentially beyond. We'll kind of see how our library of samples builds over time. I just want to note that right now we're focusing on the southern shore. We do intend to expand this to Isle Royale as well in the future, and we have some targeted areas in Isle Royale that we're looking to sample. We're still working on some permitting with the National Park Service to get that going. But so for now, we're looking along the shoreline of Wisconsin and of Michigan. All right. So we'll be sending out kits to everyone who opts into the program. The kits will include scissors for taking the fin clip. We'll have these sticker rod rules that people can use if they like. We like folks to get a fish length measurement if possible, along with the sample. An instruction card laying out the protocols and best practices for taking samples. Prepaid mailers, envelopes, stickers, pencil for doing your note taking in the field. All of that will be provided, so you should have everything you need aside from a fly rod or spinning rod or whatever you use to catch fish out there to do the sampling. So as far as taking the fin clip, we are looking for tissue from the pelvic fin down here. There's about a dime size tissue sample. And so it really helps when you're taking these samples to have your scissors ready to go. You don't want to have a fish on your line and be digging around with your scissors. It does help to have somebody with you if possible. It's not a requirement, but it can help to have a second person with you. But when we do this, you're basically you catch the fish. Ideally, it's really helpful to have a landing net if possible. Really makes it a lot easier to work over the fish assuming you plan to release it. And if it's not a legal fish to take. So you're fishing the net, grab that ventral fin in one hand and take that little dime size clip with the scissors in the other hand. Make sure you're holding on to that fin clip, and then release the fish. Again, we provide these little coin envelopes here. So each coin envelope will hold one fin clip. So when you take that fin clip, you'll drop it in the coin envelope, um, push it in there, but you'll want to leave it open for a period of time to allow the fin clip to dry out. It's really important for the preservation of the DNA for analysis that the fin clip is allowed to dry out and isn't kept moist. So, don't throw them in the envelope and toss them in a sealed plastic bag. You'll want those envelopes to get some air, and let them dry out for a day or so before you seal them and send them in. Um, so once you've taken your fin clip, you've put it in the envelope. Oh, and I forgot to mention, while you have the fish, we ask that if possible, you take a length measurement from nose to the tip of the tail to the nearest half inch. And you'll be recording that on your collection envelope. I'll talk about that here in a second. So we also like location information for the fish that you catch. If you want to provide GPS coordinates, that's great. That's preferred. But we recognize in that data, that GPS data will not be made public. It'll be used for internal use only. We may create maps, kind of like the ones that I showed you before, but they won't include very specific catch location information. It'll be much, more generalized. That said, if you're uncomfortable with providing the catch location in a GPS format, we do have some alternative options for you. Um, so we've broken the shoreline up into sampling reaches that are roughly 10 miles long along the lake shore. You can see the map here of all the sampling reaches along the study area. And what I can do is, if using a app called Avenza, I can send you this map and it will show you your location in relation to these sampling reaches. So you can simply open up the Avenza app, open up that map, look for your blue dot on the map, and see what survey reach your lake caught fish was found in. And so this is an alternative option, something you can opt into if you aren't comfortable sharing GPS coordinates. Similarly, for stream caught fish, if you don't want to share GPS coordinates, we're just asking for the name of the stream and the nearest road crossing. And you can record whatever method you choose. You'll record it on these envelope or on these pre-printed labels that will be on your coin envelopes that we provide. So for all samples, we'll ask you to put your initials. Each angler will get an ID number that we'll ask you to put on there to show who caught the fish, the measured length, the date of the catch. And then if you're in the lake and you're not providing GPS coordinates, you would put in that segment ID on this field. For a stream caught fish, same stuff up here, only you'll put the stream name and the nearest road crossing here on the label below. If you're opting to share GPS coordinates, you can leave this lower label completely blank and just write your GPS coordinates on the back of the envelope. Preferably decimal degrees, but if your GPS puts out a different format, we can always convert it. So as you accumulate your samples, we suspect that people will be out fishing throughout the summer season. You may collect a handful of samples at a time. You may choose to send them in a batch, whenever it makes sense for you. But we will provide mailers with prepaid mailing labels that will get your samples to Nadia's lab free of charge. What I've done, I've contributed to some samples to Ontario, and when I go make a trip up there, I collect all the samples from, the people I'm fishing with, myself. When I get home, I get them all organized and send them off in a batch to the lab partner there. And so that's how I recommend doing it. So generally, again, what we're asking folks to do is, whether you fish in the big lake, whether you fish in lower parts of the tributaries or both. For any brook trout you're catching in the big lake, we're looking for samples. For any fish 7 inches or larger in these tributaries, lake accessible tributary reaches, we're looking for fin clip samples. And as far as next steps, uh, we were asking that folks, we've gotten emails from a lot of folks who have expressed their interest in helping. And so after this webinar tomorrow, I'll send out an email to the entire list of folks that reached out to me with their interest, asking for folks to opt in. And so by opting in, you'll send me your mailing address, perhaps some other information that I'll request. And then I'll be putting together the sampling kits and mailing them back out to you so you can start fishing and collecting samples. It's our hope that we can gather a couple 300 samples from anglers over the next few years. I think if, at the rate of interest we're getting, hopefully folks are running into these fish from time to time. Um, we can reach that goal. Ideally, folks have areas where they have experience catching brook trout, and they can just sort of build this into their normal fishing excursions and fishing plans. If you're someone who knows nothing about coasters, is not sure where to start, we might be able to think about pointing you in some directions of places to try fishing to capture these fish. But again, the goal for a lot of this is to find these fish in places where we don't know they exist. And so we can have a conversation, with each interested angler about what works for them and try to provide what support we can to make this as successful as possible for the angler as well as successful as possible for the study. The one other thing I'll quickly mention is that these fin clips, they're non-lethal. The fish will over time regrow this tissue. So it should have limited, assuming you're practicing good catch and release methodologies, it should have minimal impact on the fish. So I think that's all I've got. And so I would welcome Nadia or Troy to chime in with anything they want to add, and then we can also open it up for questions.
That was great, Jake. Yeah, I think I'll just add that Jake's shared some of the maps before with the little pies on it where we could kind of see which streams have their own unique genetic profiles, and then that also allowed us to see where brook trout are moving from one stream to another and how they're using the lake. And I just wanted to emphasize that, without the help of anglers like all of you for that Minnesota North Shore project, some of those pies would have been missing completely, or we would just have less information on those pies. So I think, yeah, I just wanted to emphasize, I think, the role you all can play in this type of science is really important and kind of irreplaceable. And, it's often the case that we wouldn't be able to study certain populations or certain streams if we didn't have anglers out participating. So just wanted to reiterate what Jake shared earlier. Appreciate you all being here and thinking about this and being willing to take the time to consider participating. And yeah, the samples you collect, we will definitely use. They'll come to me here in North Carolina. I'll start processing them in the lab. We'll extract DNA from them, and they'll go right into our genetic workflow and keep building on the data that we already have for brook trout in the Lake Superior Basin. And then similarly, the same process will happen for the stable isotope work at University of Wisconsin. Um, and the other thing maybe I'll just add too is coastal brook trout are unique to the Lake Superior Basin, so it's pretty awesome that we have this resource here in Lake Superior. It's a really cool life history, as many of you know. These fish are beautiful. I mean, yeah, it'll just be, I think, useful to have additional information for Minnesota Wisconsin and Michigan, especially Wisconsin and Michigan, through this effort. So I just thought that worth mentioning as well.
Thanks, Nadia. Yeah, and I should mention that as Nadia said, these samples are going to be, analyzed, added to the database, and there'll be ongoing, um, I guess, interpretation of what we find over time that will be shared with, various stakeholders, management agencies, tribes, and others around the basin. And then ultimately, we hope and expect that this will end up, a published paper, down the road as well. And so you're contributing, I think, to, short-term inferences of, where these fish are to aid in management, and also a longer term understanding of coastal brook trout around the basin. And so it's some critical questions that we're answering here. And like Nadia said, we won't be able to answer them to the fullest without the assistance of anglers. Troy put in a note that was a good note. It's to help people assess whether their stream reach is Lake Superior accessible. If it has steelhead, coho, other Great Lakes salmonids in it, that means you're in a lake accessible reach. That's a good indication there. I do see a question says just email you a mailing address for the kids. That's right. Yeah. So I'm going to be asking for mailing addresses from everyone. You can go ahead and email me. I will prod everyone tomorrow. Once I receive your mailing address, I'll probably start getting kits out this week. So yeah, I just need a mailing address from everyone.
I'll build on Nadia's comments and Jake's a couple things. Jake mentioned about the fin clips regenerating, and we've clipped hundreds of fish over the years, and at sites that we go back and sample the following year. And when we clip off a little piece of fin like that, when you go back next year, you cannot identify any fish that have had their fins clipped. It all regenerates, so that's perfectly harmless to the fish if done properly. And then just adding on to how much you can contribute as anglers. It is very tricky to collect fish out in the lake for a crew. The crews that we've had sampling Lake Superior go out in an electrofishing boat at night, and there's limited access in Lake Superior, and going out at night is tricky, and it's not safe, and the weather makes it touch and go each day, so it's hard to schedule crews. And I think you could contribute a tremendous amount by providing samples from the lake. Also, our offices, we have basically 3 offices in Michigan along the whole Lake Superior shoreline, so it takes a lot of time to get to any particular access point, and we don't have ready access to many of these areas. And likewise, in the streams, our electrofishing crews on a survey, we would maybe sample 1,000 feet, but as you as anglers know, trying to find a large sized brook trout in 1,000 feet is kind of a hit and miss proposition. We can often catch quite a few smaller ones, but catching those larger fish, they're often few and far between, and an angler can cover quite a bit more water over the course of a few hours than our crews can. So it would be a huge help to us.
Thanks, Troy. Any questions out there? Folks can feel free to put in the chat or unmute yourself and ask any questions.
Maybe one thing I'll add, if you all are maybe thinking through the process, is um, collecting the fin clip sample is pretty easy, as you've seen. It almost sounds like more when you hear it described, but it's really just hooking up with your fish, um, taking a little piece of the fin, putting it in the envelope, writing the length down, writing what location information you want to share, and that's it. So our goal is to make it as easy as possible for you all, including with the prepaid mailers, through the postal service. So whatever we can do to lower the barrier to make it easy, we're happy to do.
But having a landing net really does help. So if you can have that fish in the net in the water, you can fumble around and that fish will be just fine until you're ready to take that clip.
Yeah, yeah. When I've only done this, and I've never done it in the big lake, but I've done it in streams while angling. And yeah, I'll just kind of set that fish in the net in the water, kind of lock it between my legs and get on my knees and do my quick work with, the fin clip, and then it's off to the races. So especially after you get one or two under your belt, it's very simple and easy to do. Folks can continue to reach out with questions. But one other thing I want to mention is that this is sort of our first sort of wave of outreach and onboarding, getting people involved in this. So please, but we're going to continue to bring people on board. And so please help us get the word out if you have fishing friends who might be interested in helping out with this. Our buddies that run into brook trout in the big lake or in lower parts of these rivers from time to time. Please do let me know and share, and connect those folks with me. Similarly, if you're out fishing with a group and someone else catches a brook trout, take a fin clip from their trout. It doesn't have to be a fish that you catch. If if you're fishing with a group, and yeah, we definitely want to get as many samples as we can. So just wanted to make that clear. I see a 7-inch minimum length for fish caught in streams. No, and Lake Superior. So no, if you catch a brook trout in Lake Superior, I mean, we'll take a fin clip from pretty much any size fish that you catch in Lake Superior. It can be, it doesn't matter what size it is. If it's a river caught fish or tributary caught fish, 7-inch minimum is what we're looking for. And it's because of a mixture of, we're underrepresenting larger fish in our electrofishing data set, and those larger fish are more likely to have spent some time in the lake than a smaller fish that you catch. And so that's why we set that 7-inch minimum. It also matches up with the minimum length for the minimum for keeping brook trout in Michigan. It says, how does this work complement the acoustic telemetry study conducted with the Red Cliff tribe in past years? I can maybe speak to that a little bit, but I'm going to see if Troy, how familiar you are with that before I jump into it. You don't know much about it? Okay. So I have a colleague that has worked with Red Cliff on that work quite a bit, so I'm a little less familiar with it. But my understanding is that work is primarily looking, they do some stocking of brook trout in Lake Superior and in Lake Superior accessible stream reaches, I believe. And that telemetry study, they're actually looking at where those stocked brook trout are going. So they're tagging them before they stock them, from what I understand, and trying to understand, I think they haven't been seeing these brook trout return to their streams to spawn, so they're trying to figure out where those fish are going. But I would need to check into that a little bit more to speak super specifically about it. What I can say is that Ian Harding with Red Cliff, who leads that work, is part of our brook trout working group within the Lake Superior Technical Committee and is in the loop on this work, and likely, I'm assuming, has and will continue to contribute samples to the effort. And so Red Cliff is a partner.
Paul Piszczek, do you know anything about it?
Oh, I forgot Paul was on the call. Sorry, Paul.
No, that's all right. I do not know anything about the extent of all that work.
And I'll maybe just add, I don't know about the telemetry work in particular, but as Jake mentioned, Red Cliff and their fisheries biologist, Ian Harding, have been contributing a lot of fin clips, much like Wisconsin DNR and Michigan DNR, to some of the genetic work that we've done already, which has, been quite valuable.
I will mention, thank you all for being on the call and getting involved with this project. If it becomes anything like what Minnesota has accomplished over there, I think we'll be in really good shape for some really nice information for brook trout across at least the South Shore of Lake Superior. So thank you very much. And just keep in mind too that all the the regular fishing regulations still do apply throughout this entire project. So just keep an eye on all that. Seasons, bag limits, and lengths, there's no immunity to them during the study.
Yeah, thank you, Paul. Good point there. Any other questions? Well, so as you move through this process, I'm basically your first line of tech support, I guess. If you have questions or about the study, about the methods, about anything that you're doing, I'm your person to contact with those questions. And if I don't know the answer, I'll get the answer for you. So we're here as your support team, and we really appreciate all your interest in contributing to this effort, and we're excited to see where this can go. So I think I will end it there, but no, we'll be in ongoing contact. So again, look for a message from me tomorrow. I'll request some information from you to opt into the program, and then we'll get those kits to you, and you can start fishing. So. Thanks again so much. Appreciate you taking the time tonight. And yeah, look forward to working with you on this.